Especially for this occasion, I'm going to talk about a linearized non-coding system, but at least it involves neurons. And what we did is we studied the relationships between peaks in different frequency bands in the electroencephalogram. And when Jennifer and I met some years ago, she proclaimed to be a fan of this study because it tied in nicely with what she was studying at the time. And so thank you very much, Jennifer, and the other organizers for inviting me to this workshop with eminent speakers. This work is from a while ago, and it was done together with Peter Robinson, who was my PhD advisor at the University of Sydney. Oh, are you still seeing, by the way, like on the right side, my the video extra in front of the slide, or
there's a little black box.
I should remove that.
It's smaller now.
This is better, right?
Yeah, that's good. It's gone.
Yeah. So a human EEG spectrum in the eyes closed resting state may look something like this on the left. And typically frequency bands are defined with limits that are not individualized, but that are the same across all subjects. The most prominent feature is the alpha peak around 10 Hertz, which is strongest over the back of the head, but also present frontally. And a beta peak may be present in the range from about 13 to 30 Hertz. Because of individual differences, it's not directly clear how these frequencies are related, but over the years, a number of theories have been proposed. One idea is that the center frequencies of subsequent frequency bands are separated by an irrational factor, which is equal to Euler's number or the golden ratio. So according to the latter hypothesis, for instance, low beta peaks would on average occur around 16 Hertz. And the proposed advantage of irrational frequency ratios is that signals in the different frequency bands do not interfere and can thus carry independent information. If you're skeptical like me, and you will think, but the brain is finite and noisy, so how can it possibly represent irrational numbers? But these authors show that the given frequency ratios enable the interference between rhythms to remain small, even in the presence of noise. However, this does not necessarily mean that these frequency ratios are actually the dominant ones in empirical EEGs. So, according to the second hypothesis, each peak is generated by a separate population of pacemaker neurons whose intrinsic properties make them resonate at the given frequencies. And this implies no particular relationship between peak frequencies. Then there are works that predict peaks at several non-harmonically related frequencies based on purely cortical mechanisms. Paul Nunes, for instance, distinguishes between local and global alpha rhythms, where the global rhythms are thought to partially consist of standing waves, whose frequencies are thus determined by the cortical axonal propagation speed and spatial boundary conditions. And finally, there's a hypothesis championed by Peter Robinson, in which thalamocortical resonance creates an approximately harmonic progression of peak frequencies. And this is the prediction that I set out to investigate. In other words, can we consider alpha as a fundamental frequency and peaks in the low and perhaps also beta ranges as harmonics at twice and 3 times the alpha frequency, akin to the overtones that arise when you pluck a guitar string at different positions? There was already some previous work that pointed in this direction. For instance, a study by Nicolin and Brismar showed that alpha and beta rhythms are phase synchronized as indicated by the concentration of beta relative to alpha phase at about -1 radian, which corresponds in the EEG time series to an alpha rhythm that is not quite sinusoidal, but displays little dents, as you see in panel A. To revisit this issue, we had at our disposal a particularly large sample of EEG data from healthy subjects from the Brain Resource International database. The subjects were from 6 to 86 years old. To keep things simple, we studied a single electrode, CZ, at the top of the head, which combines occipital and frontal influences. To study spectral peaks, we need to first estimate a background spectrum and subtract it. And here we opted for determining this background in a model-based manner by fitting predictions from a thalamocortical mean field model to the EEG spectra. And this balances the dual goals of having a physiologically realistic background spectrum, while at the same time not making any assumptions about the peak locations. So this is important to understand that the full thalamocortical model predicts a near harmonic progression of peak frequencies, but we only used its background component without peaks to subtract from the empirical spectrum and then determine the peaks from the empirical spectrum. This is the mean field model. It consists of 4 neural populations, an excitatory and an inhibitory population in cortex, the thalamic reticular nucleus denoted by R, and the thalamic relay nuclei denoted by S. The relay nuclei are excitatory and the reticular nucleus is inhibitory. So there is an excitatory thalamocortical loop through the relay nuclei, which is more active in the waking state. And then there's an inhibitory thalamocortical loop that passes additionally through the reticular nucleus, and that's more active during sleep. The relay nuclei receive a white noise external drive denoted by N. The cortex is modeled as a 2-dimensional sheet on which the neural activity propagates as a damped wave. And the synaptic inputs to each population are summed linearly after an axonal delay tau, passed through a low-pass synaptodendritic filter function, and finally converted to an output firing rate via a sigmoidal function, which is centered on a certain threshold voltage and with a maximal firing rate Qmax. The thalamic populations are modeled as neural masses, so without spatial extent. To investigate the rhythms generated by this circuit, one can linearize around the fixed point and see at what frequencies linear instabilities occur. The region of linear stability and its boundaries are shown in this sort of tent diagram. It's parameterized by 3 quantities that represent effective cortical, thalamocortical, and thalamic gains. For strong positive interactions in the thalamocortical loop, so large Y, alpha oscillations here. When the gain for the thalamocortical loop is strongly negative, one needs 2 passes through the loop to complete a cycle, and therefore the frequency is approximately halved and theta oscillations arise. And for strong interactions between the relay and reticular thalamic nuclei, spindle oscillations around 14 Hertz appear, which are typical for stage 2 non-REM sleep. We can thus assume that the resting eyes closed state is somewhere here, close to the alpha instability. And the fact that these positive thalamocortical interactions produce alpha in this model is not a coincidence. It happens because the total axonal plus dendritic delay for one loop between cortex and thalamus is set to 100 milliseconds. And this delay is difficult to determine from experiments. There have been some thalamic stimulation experiments, and the latency of the cortical responses was measured. But this tends to measure the shortest delays rather than the effective delay that results from the distribution of axonal transmission speeds and which is relevant for ongoing oscillations. So James Roberts and Peter Robinson, they took a different approach. And they determined the delay by fitting the model to absence seizure activity, for which there is strong evidence for coordinated cortical and thalamic involvement. Absence seizures or petit mal seizures are epileptic seizures in which the affected person stares blankly into space for a few seconds. And this analysis yielded an estimate of 80 milliseconds for the axonal part of the thalamocortical loop delay, which together with the synaptodendritic delays gives a total loop delay of about 100 milliseconds and is hence consistent with the 10 Hertz alpha rhythm being generated in this same loop when the interactions are net excitatory. And to compute EEG spectra in the model, the measured activity is assumed to be proportional to the activity of the cortical excitatory neurons, as pyramidal cells are more elongated and aligned than inhibitory interneurons. And then the signal is passed through a filter representing the cerebrospinal fluid, skull, and scalp. In the time series for the cortical excitatory activity, we recognize the same pattern that was observed in the empirical EEG by Nicolin and Brismar, with alpha activity modulated by small dents representing beta. Now comes the part where we use the model to analyze the empirical EEG spectra. We first fit the linearized model to the EEG spectra, and the model spectra are shown in magenta. And you see that the model is able to capture differently shaped empirical spectra shown in blue. Then we determine the background spectrum shown in red, which is obtained by setting the connections from cortex to thalamus to 0, thus removing any resonances. You see that the background lies in the middle of the spectrum, so it's modulated by both peaks and troughs. And then we fit Gaussian functions to the peaks and troughs, determining their location, amplitude, and width. And next, we assign labels to the peaks, which depend on the band limits defined according to the location of the largest peak. And there we distinguish 5 possible sets of band limits for subjects with the largest peak in the range 2 to 5 Hertz, 5 to 7, 7 to 9, 9 to 11, and 11 to 13 Hertz. Of these, the 2 to 5 Hertz peaks are considered to be in the theta range, and the others are considered to be alpha peaks. Remember that there were also children in the sample, which tend to have low alpha peak frequencies. And afterwards, we analyzed relationships between peak characteristics only within groups. So we're not introducing spurious correlations between peak frequencies, because within each group, the band limits are identical. The fitted model doesn't capture theta peaks, especially when occurring alongside alpha peaks, because the thalamocortical loop is modeled as either excitatory or inhibitory without the gain varying in time, or for instance, as 2 parallel loops of which the excitatory one can produce alpha and inhibitory one can produce theta at the same time. So for this reason, it's sometimes necessary to fit an overlapping theta peak and trough to properly capture the empirical spectra. If the spectral peaks are harmonically related, we expect not only their frequencies, but also their amplitudes to be related. Shown on the left are linear regressions of peak frequencies for each group separately. Predictions from 2 different levels of model simplification are shown in red. And especially the group with the alpha peak frequency in the range of 9 to 11 Hertz, which is also the largest group, shows significant positive correlations between alpha peak frequency and the frequencies of peaks in the theta, low beta, and also high beta ranges. And when interpreting the slopes of these regression lines, we have to take into account that we had rectangular sampling regions because we looked for peaks within limited ranges of frequencies. And this will tend to underestimate the slopes because you can have deviations to the top left and bottom right, but not to the top right and bottom left. So the main takeaway from this is that the frequencies are positively correlated and not exactly what their ratio is. On the right, amplitude relationships are shown. Here we can combine groups because the frequency band limits don't impose restrictions on the amplitudes we can measure. For group 4, with the alpha peak between 9 and 11 Hertz, we find that alpha peak amplitude is significantly correlated with amplitudes of peaks in the low and high beta ranges. And this also holds for all subjects combined. Alpha and theta peak amplitudes are only correlated for subjects with lower and higher alpha peak frequencies, and the correlation doesn't reach significance for the sample as a whole. We gain a more intuitive picture when we simply align the empirical spectra on the alpha peak and plot their average as a function of f over f alpha. For all subjects combined, you clearly see a little theta peak at half alpha and a beta peak at exactly twice alpha. And here we don't yet see the third harmonic in the high beta range, but a hint of this becomes visible when we only consider the 10% of subjects with the highest alpha peaks. In the bottom panels, we simply plot all the fitted peaks against each other. And there you clearly see that that 10 Hertz peaks are associated with peaks around 5 Hertz. And then you have some peaks flanking alpha because the Gaussian approximation for the peak shape is not perfect. And also sometimes they're split alpha. But then there's this line of peaks around 20 Hertz with a slope of 2 and a bit less clear but still visible, a line of peaks around 30 Hertz with where you can imagine that the slope is 3. And so in summary, we found that theta, low and high beta peaks occur on average at half, twice, and 3 times the alpha peak frequency. And furthermore, the frequencies of these peaks are positively correlated on an individual subject basis, which we were able to show especially for subjects with alpha in the range of 9 to 11 Hertz. And finally, alpha peak amplitude was positively correlated with low and high beta peak amplitudes. And taken together, these findings imply that theta, alpha, and beta rhythms are often not independent, but form a harmonic progression. And this is relevant because it says that their underlying mechanisms have something in common. One possibility is that these rhythms arise as delay oscillations in thalamocortical loops, as postulated by the mean field model I discussed. But even if different or additional mechanisms are at play, the point is that they need to generate the peaks in a non-independent manner. And correspondingly, when we relate changes in alpha or beta power to cognitive processes, we need to take into account their at least partially common origin. And finally, the harmonic peak progression suggests that for some types of EEG analysis, it would make sense to define individualized band limits based on the relative frequencies at which one expects to find the peaks. And with that, we can go back into the nonlinear world. Thank you for your attention.